Good evening, everyone. So today, as you all know, we are starting a new topic. So this is again a topic from CBSE 11, and this is a chapter where we're going to study about the plant growth and development. Okay, as a part of this, we'll be discussing what plant growth actually means. Then what are the various phases of growth that you see in a plant, and then what is growth rate, right? What is growth rate? And then once we see that, we'll move to the various factors which actually contribute to the growth. It can be external factors or it can be internal factors. Okay, so all these factors actually contribute to the growth. So all those factors that we'll list out in a few minutes, we'll study them in detail in the course of the next couple of sessions. Okay, so we'll start with, so the topic is okay. Now here we'll see if time permits, we'll cover growth rate today. Otherwise we have already allotted some time for growth rate tomorrow. Okay, so we'll start with the plant growth first. Now by definition, if we take growth, okay, if we take growth, we'll start with the definition and then I'll tell you what are the topics that we're going to discuss today. So by definition, growth is irreversible and permanent. Okay, reversal essentially means the same thing, and permanent increase in the size of an organ or a part of it. Okay, or a part of it. This is the general definition of growth. This is the general definition of growth as far as your animals or plants are concerned. However, this is not an accurate definition of growth, because if you take some cells, okay, for instance, if you take the cells in a watermelon, right, these cells, they don't actually grow as such. They don't increase in their the organ size is not increasing, the cell size is actually increasing, the cell volume is increasing. So again, if you take bacteria for instance, again there in the bacteria we can't say growth of the bacteria is actually increase in its size. To some extent yes, but the actual, when you talk about bacterial growth, we're talking about its division to form, to increase its number. So the definition of growth is a relative thing. Okay, sometimes it refers to the increase in the cell number, sometimes it refers to the increase in the cell size, sometimes it refers to the overall increase in the organ or its size of the organ or its part. Okay, now when it comes to the growth, whether it is animals or plants or anything, growth requires energy. We all know that growth requires energy, and this energy is obtained by metabolic processes. Okay, now when I say metabolic process, the various cycles that we have already studied, like glycolysis, TCA cycle, all these cycles, they ensure that you get energy. Okay, so metabolic process can be of two types, catabolic as well as anabolic. When you say catabolic, the catabolic processes are the ones where you're taking a bigger molecule, breaking down to smaller molecules, and in the process you're getting energy. Energy might come, might not come, but ultimately you're taking a bigger molecule and breaking it down to smaller molecules. That's the catabolysis or catabolic pathway. Okay, now if you are taking smaller molecules, combining them to form a bigger molecule, okay, to form a bigger molecule, that becomes a anabolic process. Okay, so metabolic process basically involves both, and both the processes require energy, and growth requires energy. Okay, so growth involves both the catabolic as well as the anabolic process. You need energy, okay, for building more components. Okay, so and for breaking down also you need energy. So in order to break them down you need energy. You get the smaller components, use the smaller components and build new things with those smaller components. So catabolic and anabolic processes are critical for the energy, and energy is directly influencing the growth of an organism. Okay, now if you take the plants, okay, the general life cycle of plants, this is what we see. Okay, let's say we start with, this is a cycle, so again it's like chicken and the egg. Okay, so you start with seeds, right, and then seed germinates, it grows, you get a plant out of it, right, and the plant again undergoes pollination. Okay, pollination is a process where the male and the female gametophytes fuse together to form the seed again, and the seeds again, this is the plant life cycle. So if you talk about the vegetative phase of the plant life cycle, okay, or we look at the events starting from pollination, we'll look at the plant growth. Okay, plant growth, okay, starting from pollination. Let's start with pollination. Okay, so what will happen in pollination? Basically, you have the pollen grain, right, which is equivalent of your sperm cell. This is sperm cell. Now this pollen grain will fuse with the ovary. And where is this ovary present? Ovary is actually present in the ovule. Okay, ovule has the, now where is this ovule present? Ovule is actually present in the flower, especially the base of the flower will actually form the, so the base of the flower, so you have your, right, you have your, the base will actually form the, so this is where your ovary is actually present. Okay, now what will happen? So this is your stigma. So when this pollen grain, okay, actually falls on the stigma will start releasing some chemicals which will stimulate the pollen grain to form the tube, and this tube will move all the way and reach the ovary or the mother cell. Okay, mother cell. Now two nuclei, okay, two nuclei will actually pass through the pollen tube and reach the mother cell. Okay, two nuclei will reach. Now in these two nuclei or these two sperm cells will fuse with a couple of components. Okay, the first one will actually fuse with the endosperm nuclei. Okay, so you have the first one fusing with the endosperm nuclei, and the second one is actually fusing with the ovary or the egg cell. So this is already diploid, so it has two nucleus, so this becomes triploid, and you have a, with the fusion of the ovary you have a diploid zygote. You have diploid zygote. Okay. So you have your pollen grain, right? The pollen grain is reaching the stigma. Okay, and this is we're talking about the general fertilization process in flowering plants. Okay, the pollen grain will form the pollen tube. The pollen tube will reach the mother cell, and in the mother cell you have the antipodal cells, and then you have the polar nuclei, and then you have the egg cell. Okay, now here one sperm will actually fuse with the polar nuclei to form the triploid endosperm, and the other sperm will actually fuse with the egg cell. Okay, so here this is where your ultimate thing, this is where your, so you have your egg cell here, and coming from there is your sperm cell or the pollen grain. Together they are forming the zygote here. Zygote is actually formed at the base of the flower. You can say at the base of the flower. Okay, now this zygote which is formed is initially single celled. Okay, the zygote which is formed is single celled. Now this single celled zygote will then develop into embryo. Okay, this single cell will develop into embryo, and in the process when this development is happening, you also see the endosperm development, okay, and also the seed development. Okay, the seed starts to develop, and the entire thing will go into to form fruit. Okay, once the pollination is done, you see the fruit formation, and when the fruit is being formed, okay, simultaneously you will see the embryo, the zygote, the single cell zygote getting developed into embryo, and you also see the endosperm forming, and finally the seed developing inside the fruit. Okay, so basically you have your seed, and inside the seed, okay, initially you had the zygote. This zygote actually developed into a embryo. Okay, so this is the initial stage. This is the initial stage. So here you have the zygote. Okay, here you have the zygote, and this is your endosperm nucleus. So endosperm is actually developing here, and the entire thing is actually in the sac called embryo sac. Okay, this entire thing is in the embryo sac. So this will develop into this entire ovule. This is ovule. This entire ovule will develop into a seed. This will entirely develop into a seed. So in the seed, when this entire thing, when the outer layer is actually developing into a seed, okay, the inner layers are also growing. This will grow into a mature endosperm. This part will grow into a mature endosperm, and this zygote which is there, okay, this zygote which is there will start to grow into a embryo. This zygote will start to grow into a embryo. So this is what happens. So if you look at this part, this is your zygote. Okay, the zygote, and this is the zygote nucleus, and zygote also has a vacuole in this particular picture. Okay, now this zygote will start to divide to finally form a embryo, and the entire process, when this is going on, this ovule is going to develop into a seed. Okay, now here, so if you take the zygote, right, the zygote will start to divide, okay, to 2-cell stage, 4-cell stage, and by the time it is in the initial stages where it is 2, 4, 8, we can say this is the pro-embryo stage. Pro-embryo stage. Okay, it's still not developed into a embryo, but the minute it hits the 16 cells range, we can say that the embryo started developing. From the zygote, you are actually seeing the embryo developing, and this embryo, this zygote to embryo development will go through several phases. Where you will see a heart-shaped transition, then you will see a torpedo transition, and finally inside the seed itself, you will see this final formation where you are seeing the formation of 2 cotyledons. Okay, this is in the early seedling. Okay, so you will see rudiment cotyledons, not developed cotyledons, very rudiment cotyledons. Okay, and then you will see the various other components, okay, like your hypocotyl, okay, and root meristem. All these rudiment, all the tissues that you will find in most of the tissues, I won't say all, most of the tissues that you will find in a mature or adult plant, you will find everything being made inside the embryo sac of a ovule, that is in the seed. So in the seed we have this ready. Okay, so this is how it is. So in the seed, this part is now grown into your endosperm. This is your endosperm, right? And this part is the zygote, which now multiplied and finally it formed the embryo. Okay, so this embryo obviously has cotyledons, okay, and these rudimentary cotyledons are what we call the scutellum. The rudimentary cotyledons are what we call the scutellum. Okay, now what you have to understand here is there's a layer outside the endosperm, and that layer is called the aileron layer. Okay, and aileron layer, okay, has some cells. A layer of cells are present in the aileron layer. Okay, so you have a layer of cells just touching the aileron layer. Okay, now what are the events that occur when a seed? So what will happen in the next phase? In the next phase, so we have the seed, right? And in this seed we have the embryo. Okay, we have the embryo which is developing. In the next phase it has to germinate. Okay, so let's see what happens in this phase of the growth. What happens in this phase? So here we are talking about the events that occur that occur during germination. Okay, so here the first thing that happens is you need water. You need to water the seeds. Okay, when the seeds receive water, okay, when they get water, water enters the seeds. Now inside the seeds, okay, inside the seeds you have a plant hormone called gibberellins. Okay, GA, short form is GA. You have this hormone inside the seeds. Okay, inside the seeds you have this hormone. So this hormone, now that the seed absorbed water, this hormone starts to mobilize. The hormone mobilizes. It starts to move. Okay, when this hormone starts to move, okay, it will go and bind to the cells near the aileron layer. It starts going and binding to the cells at the aileron layer. Now this aileron layer, okay, when this gibberellins go and bind to the cells near the aileron layer, the cells in the aileron layer will start to secrete an enzyme called alpha amylase. They start secreting alpha amylase, and this alpha amylase which is being secreted, okay, will start acting on the starch which is stored in the endosperm. Okay, the endosperm basically has starch stored in it, and the alpha amylase will start acting on the starch, and when the alpha amylase acts on starch, the starch is broken down to glucose. Okay, this glucose is then used by the developing or growing embryo to start the growth. It starts to grow. Okay, so basically all these events are occurring, okay, because the embryo needs energy for the growth, and in order to get the energy you need glucose. Okay, the raw material here is glucose. Now how do we get the glucose? The glucose is actually stored in the form of starch. Okay, it's not free, so the starch has to be broken. In order to break the starch you need to mobilize gibberellins. So the reason why we water the seeds is, when you give water to the seeds will absorb water. The gibberellins inside the seeds will start to move. They move and go and bind to the cells in the aileron layer. These cells will start to secrete the enzyme alpha amylase. Alpha amylase will go and act on the starch which is present in the endosperm layer and endosperm region, and in turn the starch is broken down to glucose. Glucose provides the energy for the embryo and it starts to grow. Okay, so let's look at the events very quickly. So you can see here the water is getting in, and this water will start the mobilization of gibberellins. The gibberellins will start to move, and they go and bind to the cells in the near the aileron layer. Okay, they go and bind to the cells in the aileron layer, and these cells, when they bind to the aileron, when the gibberellins bind to these cells, they start secreting the alpha amylase. Okay, here it is written as enzyme secretion, and the enzyme that is secreted is your alpha amylase, and this alpha amylase will act on the starch which is stored in this region, the endosperm region, and as a result you start getting glucose. Okay, the glucose is released. The glucose is then utilized by the embryo. The glucose is utilized by the embryo and that energy is used for the growth. Okay, and that energy is used for the growth. Basically, the embryo has two components at this stage. At this stage, it has two components. One is cotyledons, and the other component is your embryonic axis. Okay, now from one end of the embryonic axis, okay, two components. One end of the embryonic axis, you will see the development of radicle. Okay, from one end of the embryonic axis, you will see the development of radicle. Okay, now this radicle will ultimately form the root system. Okay, now the cotyledons on the other end, okay, are bound to the plumule, so which is present on the other end of the embryonic axis. This plumule will start to grow into the shoot system. Okay, will start to grow into the shoot system. The cotyledons are actually attached to the other end of the embryonic axis, and that part is, so here this germination process, this is the mechanism involved in germination process, where you need the gibberellin's movement for activating the cells of the aileron layer to start releasing the enzyme alpha amylase, which is required for the release of glucose from the starch, and that is used for that is used as energy for the growth. Okay, now this germination process can occur by two ways. The germination process can occur by two different methods. One is called the hypocotyl germination, and the other one is called the epicotyl germination. Okay, now in hypocotyl germination, okay, in hypocotyl germination, we see that the cotyledons actually come out of the soil surface. The cotyledons, okay, come out of soil surface. In epicotyl germination, the cotyledons remain in the soil. Okay, what do I mean by that? So this is the hypocotyl germination. So you see the seed, inside you have the embryo, right? Water, it absorbed water, then the gibberellin started moving. Okay, it started providing, it went and bound, it went and then started stimulating the cells in the aileron layer, near the aileron layer. They started secreting the amylase, glucose was released and the glucose was used. Okay, and the water did another thing. The water actually caused the cells, the seed to rupture. Okay, after absorbing water, the seed swells up, as a result it ruptures. Okay, and from one end of the embryonic axis, you see the development of radicle. Okay, and from the other end, you see the cotyledons being pulled out, okay, with the help of the hypocotyl. Okay, these are actually being pulled out of the seed coat. Now, once they are pulled out of the seed coat, you see the cotyledons actually coming out above the soil surface. Okay, these are your cotyledons which are actually above the soil surface, and the radicle will then grow into the root system. Okay, then you have the hypocotyl and epicotyl finally growing into the shoot system. Okay, this part is actually forming the plumule from where your shoot system is coming. Okay, so here the important part here is the cotyledons are actually coming out of the soil. Okay, this can be seen in, as the example here given here, it can be seen in plants like garden bean, pumpkin, etc. Okay, however, in plants like maize, you don't see the cotyledon coming out. The cotyledon will stay or will remain below the soil. Okay, the lower part of the embryonic axis will still give the radicle. The upper part is something called the coleoptile, which will directly start giving the foliage leaves. Okay, from this you will directly see the foliage leaves forming. Okay, so this is the epicotyl type of germination, or epicotyl, epigeal, sorry, not epicotyl. This is epigeal germination. Okay, I'm sorry, let me just make the correction. This is hypogeal and this is apigeal. Okay, let me just make that correction. Hypogeal and apigeal germination. Okay, so apigeal is cotyledons remain below and hypogeal is cotyledons above soil surface. Okay, so these are the two types of germination that we'll actually see in the plants. Okay, so that is how the plant starts to grow. That is how the plant starts to grow. Now, there's a major difference between the growth in plants and the growth in animals. Okay, how is the growth in plants different from the growth in animals? Now, if you look at the growth in plants, if you take the, for instance, let's take the animals. Animals, they develop into if you take, suppose if you take dogs. Okay, now if you take one particular species of a dog, they all look or they all resemble more or less the same. They have similar shape, they have similar size, more or less similar shape and similar size. Okay, we don't see that in plants. Okay, in plants the growth is indeterminate. Okay, you will see indeterminate growth. You will see indeterminate growth in plants. Now, the reason for indeterminate growth in plants is because the growth of the plants is influenced by environmental factors to a large extent. Okay, it's influenced by environmental factors to a large extent. The amount of light they get, the amount of water they get. Okay, all these factors actually influence. When you keep two plants side by side, one in shade and the other one in let's say with enough sunlight, okay, the one with the sunlight will grow well when compared to the, even though they are from the, they belong to the same species, you have those differences because of the difference in the environmental conditions. Okay, so we say plant growth is indeterminate because you can't actually determine how it will turn out. Okay, now the other factor is in plants the growth is unlimited. Okay, theoretically it is unlimited. They divide and they continue to grow till they die. Okay, till they die. Okay, unlike in animals, the plant don't actually stop the growth. And the reason for this is they have the meristems. The reason for unlimited growth is the presence of meristems. Okay, they have the meristems because of which they are able to show unlimited growth. Okay, now, so here all the tissues in the plant, okay, the various tissues in the plant or the various types of cells in the plants can be grouped into two categories. All the types of cells in the plants can be grouped into two categories. One is meristem cells and the other is differentiated cells. Meristem cells and these meristem cells are the reason why you have unlimited growth, not the differentiated cells. I'll tell you what the difference is between these two. Okay, so basically you have two kinds of cells in the plants. All the cells in the plants can be grouped into two categories: meristem cells and differentiated cells. Now these meristem cells are like the stem cells of the animals. Okay, meristem cells are like the stem cells of animals. They're like the stem cells of the animals. That is, these meristem cells can divide on their own. Okay, and they can differentiate or change into any form they like. So these meristem cells has the ability that they divide on their own, okay, and then they can differentiate. They have the potential to differentiate into any kind of cells, and these meristem cells have very rapid growth. Okay, now let's see where these meristems are actually present. These meristems in the plant, you have two regions where you will actually find the meristems. Okay, meristem cells. The first one is called the apical meristems. The first one is called the apical meristems, and the second one is called lateral meristems. Okay, apical meristems and lateral meristems. Now, what are we talking about? We're talking about the two kinds of cells that are present in the plants are the meristems and the differentiated cells. Right? We are talking about where these meristems are actually present, and the property of these meristems are they are like the stem cells of animals. That is, they can multiply on their own, okay, and they can differentiate into any kind of cell that they want. Now, these apical meristems, if you take the apical meristems first, these apical meristems, you will find these apical meristems at the tip of roots, shoots, and branches. Okay, only at the tips. As the name suggests, apex means tip. Okay, in Latin, apex means tip. So they can be found only at the tips of the roots, shoots, and the branches. Okay, and if you look at the characteristics of these cells, okay, these cells, like I said in the beginning, they divide rapidly. Okay, they divide rapidly, and this division is what is responsible for primary growth of the plant. Okay, so the growth that is caused by the apical meristems, okay, the growth that is responsible, that is the result of apical meristem division, is called the primary growth. Okay, this is the primary growth. Now this primary growth leads to two things. One is, because of the primary growth, since these are actually presented the tips like this, so these are actually, so this is your shoot tip. Okay, since these are actually present at the tip of the stem, okay, let's say at the tip of the stem, since they are actually present at the tip of the stem, the growth will actually increase the height. Okay, so this primary growth is responsible for, primary growth leads to height or length of the shoot or root. Okay, so if it is present at the tip of the stem, this is your stem. If the meristem is present, obviously the meristems, apical meristems are present at the tip. So this meristem which is present at this tip is responsible for increasing the length of the shoot, and the other way around, okay, the meristem which is actually present at the tip of the roots is responsible for the length of the root. Okay, the root will actually grow because of the apical meristem present at its tip. Okay, now it is also responsible for development of specialized plant like leaves. Okay, the apical meristems are also responsible for the development of leaves and buds, specialized organs like leaves and buds. Okay, so that is about the apical meristems. Then if you look at the lateral meristems, these lateral meristems are actually organized in a radial fashion. That is, so if this is the stem of the plant, okay, they are organized in this fashion, okay, in a radial fashion, in a circular fashion. Okay, so this entire thing will proceed like this. So the lateral meristems are actually arranged in a radial fashion. Okay. Now these let many such concentric. So one layer here, one layer here. So several layers of meristematic cells are arranged in concentric circles, and these concentric circles are actually responsible for, these concentric circles actually form the lateral meristems. So if it is present at the tip, we call it apical meristem, and if it is present in the shoot, okay, in concentric circles, if it is arranged in concentric circles in the shoot or root or wherever, we call them lateral meristems. Okay, now these lateral meristems, they extend through the entire shoot. If this is stem, they'll extend entire like this, okay, or if it is root, they'll extend the roots as well, the entire root as well. Okay, there'll be extensions of these lateral meristems all through the shoot, all through the root as well. Okay, and these these lateral meristems, one important that you have to understand here or remember here is these lateral meristems are found only in the dicotyledon plants. Okay, you don't see the lateral meristems in the monocots. Okay, you see them only in the dicots. And these lateral meristems are the ones which are actually responsible for secondary growth. They are responsible for secondary growth is because of the lateral meristems. Okay, so the secondary growth, because of the secondary growth, you see increase in the diameter of the roots and shoots. The diameter will actually, so with the apical meristems, you see an increase in the length of the plant. Okay, it can be the shoot or the roots, and with the lateral meristems, you see the secondary growth. With the apical meristems, you are actually seeing the primary growth. With the lateral meristems, you are seeing the secondary growth. Primary growth is responsible for the height of the plant, and lateral meristems are responsible for secondary growth, and secondary growth is the reason why you see the increase in the diameter of roots and shoots. Okay, and yeah, and this secondary growth, you won't find this in all the animals because in all the lower plants. You will see the secondary growth only in higher plants. Okay, and this secondary growth can be seen only in higher plants like for instance your pines, okay, or any other woody plants like conifers, anything. Okay, all the woody plants, the wood is because of the secondary growth. The wood formation is because of the secondary growth. So in smaller plants, okay, which do not, with no secondary growth, in plants which are small with no secondary growth, they are weak and they have soft bodies. So if you come across a plant which is actually weak, which you can actually easily bend, okay, the reason why you are able to do that is it lacks the secondary growth. And the reason why it lacks the secondary growth is it lacks the lateral meristems. Okay, so if it is able to grow taller, then we can say that it still has apical meristems. But if it is not strong enough, if it doesn't have the secondary growth, that is, it doesn't have the woody structure, then we can say that it lacks the lateral meristems. And like I said in the beginning, all the monocots lack the lateral meristems. You won't find the lateral meristems. So that's the reason why your maize and all will not form the wood. They have very weak and soft bodies because they lack lateral meristems, which can actually form the secondary growth, okay, which gives a strong support to the plant, okay, which makes it hard, okay, which makes the stems and roots hard. So since these organisms lack these lateral meristems, they have very soft bodies. Okay, now the growth is, I told you in the beginning that growth can be relative. Okay, so it can mean different things in different situations. Okay, so whatever is the situation, growth can be measured. Growth can be measured. Okay, so since we cannot measure the, for instance, to be specific, if let's say is now increased, here it is increased. Okay, it's hard to measure the increase in the protoplasmic volume, okay, of the cells. So whether the inner contents has increased or not, it's hard to measure that. But we can actually measure a few components which will tell us whether the overall cells size is increasing or not. For instance, let's say we took a plant, okay, and then we measured its weight, okay, fresh weight, and after a few days you measure the fresh weight again, okay, after a few days you measure the fresh weight again. If the weight of the plant is increasing, then we can say it's actually growing. Okay, so there are many ways that you can actually measure the weight. We just have to go around to actually measure the weight. So the various things that you can do to measure the growth of a plant are measure the fresh weight of the plant, or measure the dry weight, right, or measure the length, okay, measure the length, and if it is, let's say, woody, measure the area, okay, how much actually area is it actually covering, okay, then the volume, okay, and cell number. Cell number can also be measured. Okay, so all these components will tell you whether a plant, so plant growth can be measured in terms of this. For instance, if you talk about cell number, okay, if you take one root apical meristem, okay, of maize, if you take one cell of maize root apical meristem, okay, it divides to give you 17,500 new cells per hour. So it divides so rapidly. So here the growth, if you have to talk about the growth of this particular root apical meristem cell present in the maize, we can talk in terms of the number of cells it is actually producing per hour. Okay, it's the increase in the number of cells that we are referring to as growth. Okay, here growth means the increase in the number of cells. However, if you take watermelon, okay, it doesn't actually divide at such a higher rate. If it doesn't the cells of watermelon will not divide at such a higher rate. However, the size of a watermelon cell, okay, watermelon cell size increases by 3,50,000 times. Okay, so the size here is increasing. So when we are referring to watermelon cell, the growth here is in terms of the cell size. The cell size will actually increase by 3,50,000 times. Okay, so growth can mean different things, and we can measure different things in order to know whether a particular thing is growing or not. In the case of maize root apical meristem cells that we just discussed, it's the number of cells that is telling us whether the root apical meristem cell is dividing or not. And here in the case of watermelon, it's the cell size which is actually increasing. It's not dividing to give new cells, it's the cell size which is actually increasing, and it's increasing to a large extent. Okay, now we'll look at the phases of the growth. Phases of the growth. What are the various phases of the growth? Now, the period of growth in a plant, okay, the period of growth in a plant can be divided into three components, okay, three phases. One is meristematic phase. Then we have the elongation phase, and finally we have the maturation phase. Okay, we can say the cells, okay, the growth phases, you can say you will find three different kinds of growth phases in the plants. One is the meristematic phase, the elongation phase, and the maturation phase. This can be well explained if we take the root structure as an example. The root structure as an example. Okay, let's take the root structure as an example. Okay, now here we can see the lowermost zone is the meristematic zone. Like I said, you have the apical meristems towards the tip of the root. Okay, so here you can see all the cells are of the same type. All the cells are of the same type. They are not differentiated. They are not converted into a different form. They all look the same. Okay, they all look the same. And here these cells, okay, the cells of the maturation phase, the cells in the maturation phase have few characteristics. Okay, the cells in the maturation phase has few characteristics. One is they have rich protoplasm. They have rich protoplasm and they divide very rapidly. They divide at a maximum rate. Okay, they divide very fast, very quickly. I just gave you an example of maize root apical meristem, which divides to produce 17,500 cells per hour. Okay, so that is the rate of divisions that you will actually see in such meristematic zones. Okay, in the cells which are actually in the meristematic phase, you will see the division in that fashion. Okay, now here another important feature that you have to understand or remember here is the cells of the meristematic zone, or the meristematic the cells which are actually going through the meristematic growth phase. Okay, they have very thin primary cell wall. They have very thin primary cell wall and it's deposited with cellulose, and they are interconnected very well with plasmodesmata. You will see lots and lots of plasmodesmata connection. So you just have to remember one thing: the cells in this zone divide very rapidly. They have rich protoplasm and they have very thin cell walls with less deposits of cellulose. So these are actively dividing cells. Now right next to this zone, okay, right next to this zone we have the elongation zone, where you will see the cells which are in the elongation phase. You can see the cells here. Let's say the cell here looks like this. In the elongation phase, you will actually see the cell elongating. Okay, you will actually see the size of the cell increasing. You can see here, this is the size of the cell here. Okay, the cells which are in the meristematic phase, they are smaller when compared to the cells in the elongation phase. Okay, the cells of the elongation zone are actually in the elongation phase. They, as the name suggests, they elongate, that is, they increase the cell size. Okay, the cell size will actually increase, and here you will see lots and lots of vacuoles in them. One reason why the sizes increase, and you will actually see new cell wall being deposited here. Okay, new cell wall gets deposited in the cells which are in the elongation phase. New cell wall gets deposited. Okay, now if you move further up, you will see the maturation zone, where you will see the cells in the maturation phase. Now what do we mean by maturation? So like I said in the beginning, you have meristematic cells. Okay, and these meristematic cells has the ability to differentiate or convert into any kind of cell they want. Once they convert, we call them the differentiated cell. And these differentiated cell will perform only one function. Okay, they are differentiated to perform a particular function. For instance, if you take humans as a best example, you have the hematopoietic stem cell in the bone marrow, and from this hematopoietic stem cell you are getting, in fact, all your RBC, your WBCs, that is your macrophages, monocytes, mast cells, eosinophils, basophils, everything is actually coming from the and the blood platelets are also coming from the same component. Okay, so all these cells are coming from one cell. This is the stem cell, and this stem cell can differentiate into any kind of cell, but once they are converted into the RBC, they can perform just one function, which is transport of oxygen or other materials. Okay, WBC can perform the immune activity or immune functions. Platelets can perform the clotting functions. These cells, the blood platelets cannot suddenly go back to form the RBC and start performing a different function. They are committed, they are differentiated, they cannot go back to form any other kind of cells. But these cells, they can actually form in, they can divide on their own and they can form into any one of these cells. Okay, so in the meristematic zone, you will see the cells which are undifferentiated, okay, and they can divide, and once this tip starts to grow, these are actually moving, the cells are actually moving into the elongation zone. Okay, in the elongation zone, what is happening? They are elongating, they're increasing in size, new cell wall is getting deposited, and when they move further in the maturation phase, you will see a few characteristics, okay, like the cells have the maximum size here. In the maturation zone, you will see the cells have maximum size here. Okay, they have the maximum size, and they have the cell wall completely deposited in them. Okay, they have thick cell walls. So these are completely matured. These are committed. Okay, so these are the three different phases of growth that you will see in the plants. The first phase is the meristematic phase, where you will see the cells actively dividing, and then you have the elongation phase where they are not dividing much, they are simply increasing their size, and then the wall deposition will start. And if you go further into the maturation phase, in the maturation phase they have elongated, or they got the maximum size, and now you see the deposition of the second cell wall materials, and several other modifications actually occur once the cells enter the maturation phase. Okay, so that is about the various phases of the plant growth. Then you have the growth rates. Okay, growth rates. Now, so if you have to define growth rate is increased growth per minute. Okay, so in short, it is increased growth of anything. It can be a plant, it can be bacteria, it can be human beings, anything. Increased growth per minute, sorry, not per minute, you can say per unit time. It varies. Okay, if you take bacteria, we can say in a minute. Okay, if you take because they divide very rapidly. Okay, and if you take human beings, obviously it will take a lot of time. So unit time, it can be minutes, hours, okay, to months, years. Okay, so increased growth per unit time. That's the definition of your growth rate. Now, an organism or so this growth can occur by various ways. Okay, so basically growth involves cell division. Like I said at the beginning, growth involves cell division or increase in the cell size. Both are. Now, if it is cell division, this cell division, okay, can occur by two ways. Okay, this growth which involves formation of more cells, okay, growth which involves formation of more cells can occur by two different ways. One is arithmetic and the other one is geometrical. Okay, two different ways your growth rate can increase. Okay, formation of more cells can occur by two different methods. One is arithmetic method and the other one is geometrical method. Okay, in arithmetic method, this is what happens. Okay, so here we have one cell which is dividing. This is single cell which is dividing, and this cell, when it divides, so you have one cell, okay, this is dividing, and when it divides, the new cell which is formed is becoming the body cell. Okay, the new cell which is formed is becoming the body cell. The old cell will again divide. The old cell will again divide, and this will again become the body cell. Okay, so single cell will divide and keep adding more and more cells. Okay, so all the cells are derived from a single cell, in short. So you see here, the single cell is dividing and giving a body cell. Again, this half is actually growing in size. This is dividing and then forming another body cell. Again, the single cell, the single blue cell here is dividing to give more and more body cells which are light blue in color in this particular picture. Okay, so from a single cell you will see the formation of many different. One daughter cell will continue to divide and it forms many cells. This is your arithmetic. This is one way where your cell number can increase. And the other way is the geometric manner, where single cell will divide into two. Each of the cells which are coming from this single cell will then divide into, so basically we'll start with two cells. So you will start with one cell. Okay, this one cell will divide into two, and these two cells will then divide into four cells, and four cells will divide into 8 cells. So this is your arithmetic. So this is your geometric fashion of cells. Okay, so division can occur by this way or this way. Okay, now arithmetic model of division can be actually seen in the elongation zone. Yeah, in the root elongating at a constant rate. In a root which is elongating at a constant rate, you will see the arithmetic mode of growth rate. Okay, so here you can see in an arithmetic mode of growth rate, you will actually see the linear increase. Okay, so with time the number of cells will be gradually, it's a linear graph. Okay, and if you have to, since this is in the graph form, we can actually express this mathematically as well. So in the mathematical fashion, if you have to express this, it is given as where LT is the length at time t and LO is the length at time 0, right? And R is growth rate or elongation per minute. Growth rate or elongation per, again, unit time. Okay, now here let me just come back to this. I hope you can see this. Yeah, so this is the time. We are plotting time against the height of the plant. We're plotting time against the height of the plant. Okay, so it can be the length of the root. Okay, in this particular situation. So here LT is the length at time. Okay, time t. So LO is the initial. Let's say you measured the length at 1 o'clock. Okay, and then let's say the next day at 1 o'clock you are again measuring the length of the plant. So this becomes time 0. Okay, this becomes time 0 and this becomes time t. Okay, this becomes time t. So this is, let's say, 1 AM and this is 1 PM, for instance, 12 hours. Okay, so this becomes time 0 and this becomes time t. Okay, and what else? Then growth rate, okay, can be measured using this particular equation. Okay, this particular equation, this particular mathematical equation can be used for determining the growth rate. Okay, now in the geometric growth we see 2 becoming 4, 4 becoming. So here the geometric growth, unlike your arithmetic growth, arithmetic growth was linear, you have a straight line. Geometric growth, on the other hand, is not linear because, okay, the growth will occur rapidly in the young plant. Okay, the growth will occur rapidly in the young plant. 2 will become 4, 4 will, all the cells will divide very rapidly. But the minute the plant matures, the growth will slow down. Okay, the growth will come down. So here the graph will be sigmoidal. Okay, the graph will be sigmoidal. So this is how you will see, and this is the graph, not just applicable to the plants. This is a growth curve for human beings, bacteria, plants, any living organism that you can think of. Okay, we all start at a point close to 0, and then we start to grow. Initially it takes time for the growth to kick in because we are getting used to the new conditions. Okay, and whether it is the plants, whether it is the bacteria, whether it is human beings, there will be some lag phase. Okay, some period where the growth is not that good, but then very rapid growth phase will be there. Okay, very rapid growth phase. So this is called the exponential phase or the log phase. Okay, your cells are rapidly dividing, you are young, your cells are rapidly dividing. Okay, you have all the necessary components that will actually support the growth. However, after a certain point, when the plant actually matures, okay, or in human beings, when the growth hormones actually stop, okay, you won't see the growth happening, you will see a plateau. Okay, so this is basically a sigmoidal curve. Okay, so even the plants will show a sigmoidal curve, and all the regions which divide by all the other tissues that are there in the plants, okay, or all the organs in the plant, or all the cells in the plants will show a similar growth curve. Okay, they'll show an initial lag phase, the growth will be slower, then rapid mitosis will happen, very quick growth will occur, which we call the log phase. Then here, because of lack of nutrients, okay, lack of nutrient supply, okay, is one main reason why you see no growth. Okay, it can be many other reasons. Okay, and once this happens, and finally death will occur. So this is the growth curve of most of the cells in a plant or organs in a plant or the plant itself. The growth initially will be slower and called the lag phase, and then rapid growth can be seen till the plant completely matures. And once the plant matures, you will see that the growth is not increasing. Okay, it's just flatlining, and finally leads to the death of the plant. Okay, so this is the growth curve of a plant, and this growth curve can be calculated using this expression. Okay, where W1 is the final size. Now when I say final size, it can be weight of the plant, final weight of the plant, or it can be height of the plant, or it can be number of cells. Okay, it can be number of cells. Then WO is called the initial size of the plant. At the beginning. Okay, at the beginning. Then R is the growth rate. R is the growth rate. T is the time of growth. And E is base of natural log. Okay, so here R is relative growth rate, and is also called, and is the measure of the ability of the plant to produce new plant material. Okay, R will tell you whether the plant is able to produce new plant material or not. Okay, by calculating R, you will know whether the plant is able to produce new plant material or not. Okay, so that is about a brief introduction about plant growth and plant growth rates. Okay, tomorrow we'll look at some more aspects of growth rate, and then we'll talk about differentiation, okay, in detail. And in the subsequent lecture we'll talk about the external factors, the internal factors which are actually responsible for the growth of the plants. Okay, so some of the doubts that were put by you are, what is zygote nucleus? So basically you have the sperm nucleus, okay, which is the pollen nucleus. It is actually fusing with the egg cell nucleus. Okay, these two are egg cell nucleus to form the zygote. So two nucleus are coming together to form a single nucleus, which is the zygote nucleus. That's the nucleus of the zygote, that's all. Okay, nucleus of the zygote. Now what is pro-embryo stage? Okay, so initially what will happen, zygote is single celled. Zygote is single celled. Now this single celled zygote will start to divide. Okay, when it divides, the single cell will become 2 cells, 2 will become 4. Okay, so these initial stages where your zygote is not completely developed, those stages are what we call the pro-embryo stage. Okay, the minute it starts to move to 16 cell stage, you can say, okay, this is an embryo which is actually growing. You can say it's a growing embryo. Okay, so all those stages before that we call them pro-embryo stages. Okay, then what are cotyledons? Right, so basically cotyledons are the ones which actually grow from the embryo. Okay, the embryo starts to divide, cells will start to divide and finally form, okay, these cotyledons structures which will actually grow in the future to form the first leaves of the plant. Okay, so that's your cotyledons. Now how do they bind gibberellins? Okay, gibberellins are basically plant hormones. The question was asked by someone, how do gibberellins actually bind? So these are plant hormones, right? So basically all the cells on their surface have many different kinds of receptors. Okay, so these receptors can actually bind many different kinds of molecules, and here in our case the gibberellins will go and bind to the receptors here, and the binding to these receptors will cause some chemical changes inside the cell which will ultimately stimulate the DNA inside these cells, and the DNA will start making RNA, and RNA will start making protein, and that protein is what we see as alpha amylase. That protein is nothing but your alpha amylase which is released out. Okay, so that is about why that is about how the gibberellins actually bind to the cells. So they bind to the specific receptors on the cells, and all the cells have these receptors on their surface. Okay, now yeah, then how is apical meristem related to primary growth? Okay, so it's not related to primary growth. What we are saying is all the growth, okay, all the growth that is resulting from apical meristems, is what we call the primary growth. Okay, and primary growth is responsible for increase in the length of the root or shoot or branch. Okay, so primary growth is actually happening at the tip of the shoot or tip of the root or tip of the branch. Okay, so if you take a plant, right, at the tip of the branch, at the tip of the root, at the tip of the shoot, you will see growth happening, and that growth is called the primary growth, and that growth is because of the apical meristems. Okay. Yeah. So what is the optimum temperature? Okay. So there are many factors that affect the growth of the plants. Okay. And we'll discuss the factors tomorrow, but since you asked a question, I'll tell you the optimum temperature is 20 to 30 degrees centigrade. For most plants, it is 20 to 30 degrees centigrade. That's the optimum temperature for plant growth. Okay. So that's all for today. So we'll meet tomorrow around the same time and continue with our discussion. So keep following us on Facebook, YouTube, and subscribe on our YouTube channel as well, and try our app. There are many interesting aspects on the app which you should be familiar with. Okay, so see you tomorrow.